If you have your lab manual, go ahead and get yourself a pen or a pencil, and we'll start recording some data, okay? And remember to open up your chat feature so that as I'm talking, if you have any questions or comments, we can record them there. Anna, Katie, Keisha, Victoria. How's he doing, Victoria?
He's doing a lot better. He has a lot more.
Hi, sweetie.
Can you say hi?
Oh, baby.
And we just did his nebs while we were on break.
Oh, good.
Oh,
Oh, I'm so glad you're home again.
I know.
Ugh.
There's cesspools. Yeah, we're glad to be home.
Nobody wants to be in the hospital.
Yeah, just the idea, huh? Oh.
Oh my goodness, I saw that one coming. I did, I saw that one coming.
Oh. Oh. You good? Yeah.
That was good. You did a good job on that one.
Can you look and see who's talking to you? No.
Hi, sweetie. Hi. Hello.
Oh.
Get that out of there. Get that stuff out of there.
Yeah, dude. All right, let's go find some things for you to do. Hello, little boy.
Hello, dear. Have you given up? Have you given up, Lucy? I'm sorry, I can't hear you. You're muted.
My back was hurting.
Oh, I'm sorry.
But I'm fully awake.
Hooray! And I know you're just itching to talk about fermentation, and that's what we'll do.
I like fermentation.
All right, let's see who's here. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 6, 7, 8. I think we're missing. No, I think that's it. All right.
Hey, everybody.
All right. So if you have your lab manual, if you have it with you, go ahead and grab it and grab a pen or a pencil before you lose me, because you just might, you never know. All right, so what I want to do now is open up the lab manual to page 121. This is the lab report for the fermentation lab. So if we look at what we'll look at now on the slides, on the pictures, is our results. We'll use these results to answer the questions in the manual. Okay. So on page 121, there's a table to record results. And they've got 4 organisms listed for us to test. We're not going to be looking at these 4 organisms. We're going to look at different organisms from this. We will look at E. coli, but as you'll see in a minute on the slide, the second organism that we're going to look at is called Klebsiella. The third one is Salmonella. And the fourth one is Pseudomonas. So if you have a pen, go ahead and jot that into your table. E. coli, Klebsiella is the second one, Salmonella, and Pseudomonas. If you don't worry, you can do it later because the names of these organisms are in the slides that are up on Canvas for this lab. Now, if you look where it says hypothesis or hypotheses on page 121, the first thing it asks is, or it states is that all bacteria will ferment the 3 sugars. So jot down for yourself that we're testing glucose, lactose, and sucrose. Those are 3 very common, simple sugars that we offer to microbes to see if they can ferment them. And we know for most of the common pathogens what they can and can't ferment in that list: glucose, lactose, and sucrose. So if we think about that statement, all bacteria will ferment the 3 sugars. The answer for that would be disagree, because as I said a few minutes ago, different microbes are incredibly variable in what they ferment and what they don't ferment. And we actually take advantage of that diagnostically. We can look at which sugars they can and can't ferment, and that's part of our data for identifying unknown microbes, okay? So the second statement for hypothesis says, a bacterium that doesn't ferment glucose will not ferment lactose. That's another disagree. Bacteria really differ. Microbes really differ. Some of them can ferment lots of sugars. Some of them can ferment only 1 sugar. Some of them ferment weird, combinations of sugars. It depends on the microbe. All right, now in the table at the bottom of page 121, you'll notice that there's room to record data at 24 hours and 48 hours. You can go ahead and cross off where it says 48 hours. We're only going to collect 1 data set for these 4 organisms, and it's going to be at a 24-hour read, just to make it easier for us. All right? So let's take a look at our results. Give me just a minute and I'll get the slide screen back up for you. Okay. So you can see at the top of this slide, it says carbohydrate fermentation, glucose, lactose, sucrose. So I've got 3 tubes that I've inoculated, and it says results. So anywhere on the slides that it says results, those are going to be the results that we analyze. So the first results are for E. coli and for Klebsiella. So you have to imagine that we took 4 carbohydrate fermentation tubes for each organism. The first tube we're just going to use as a control. We're not going to put any bacteria in it. We're going to keep it the way it came out of the box. We're going to put it into the incubator, and then we're going to use it as a comparison, as a control when we read the results at 24 hours. The second tube, we're going to put glucose in. The third tube, we're going to put lactose in. And the fourth tube, we're going to put sucrose in. We're going to make one set for each organism. And then we're going to inoculate our tubes. We're going to put some E. coli in here, and in here, and in here. And we're going to get it into the incubator. And then we're going to do the same thing for Klebsiella. Inoculate our tubes, get everybody into the incubator, and then read it at 24 hours. So what you're looking at here is the results for both of our organisms. We got identical results for the E. coli and for the Klebsiella. So remember, this is our control tube over on the left. This is an uninoculated carbohydrate fermentation tube. The broth inside is a media broth, just a growth media, a basic nutrient broth. And we have supplemented it with these sugars. So it stays red. It comes in the box red because of that indicator dye that's in there. And what we're looking for is a color change. We're looking to see if the media turns yellow, because if this organism can ferment this particular sugar that we put in, it's going to make an acid from that sugar, and it's going to turn the media yellow. And remember, the other thing we have to do is we have to look for gas inside that upside down Durham tube. So take a look at our results. In the tube that's labeled B, that's our glucose tube. Obviously, it's yellow. So both E. coli and Klebsiella were able to ferment glucose. And if you look, there's a little bubble of gas in the top of the Durham tube. Both of these organisms ferment glucose and produce gas in the process. We would record that as acid plus gas. Now, if you go over to tube C, that's our lactose tube, and it looks exactly the same. And in fact, tube D looks exactly the same for sucrose. So we have two organisms here, E. coli and Klebsiella, that are capable of fermenting a range of carbohydrate. These are organisms that we would call non-fastidious. Non-fastidious. In other words, they are not picky. They are capable of fermenting a range of sugar, and they produce gas in the process. They use a pathway of fermentation that also produces a gas. So in your data table, you don't have to do it right this second if you don't want to, but if you can, go ahead and jot down in your data table. Again, the first organism is E. coli. Under where it says glucose, did something grow? Yes. What was the color? Yellow. Was there acid? Yes. Was there gas? Yes. And if we go over to the next column, that's the lactose results. Same thing, growth, color, acid, gas. And then the sucrose result, growth, color, acid, gas. So that's the E. coli. And then the next organism down, remember you crossed out Enterobacter and you wrote in Klebsiella. Here's a little trivia for you. Klebsiella, the organism called Klebsiella, used to be called Enterobacter. Many years ago now, a group of the Enterobacter bacteria started to evolve on a slightly different path. And so we had to give them a new genus. We had to give them a new name, a new scientific name. And that's where Klebsiella came from. They used to be part of the Enterobacter, but now they have emerged on their own evolutionary pathway as Klebsiella. So you'll record the Klebsiella results identical to the E. coli results for those three sugars. Now let's take a look at the Salmonella result. Now, they're not showing us our control tube here, but we always run a control. Anytime you do carbohydrate fermentation, you tuck an uninoculated tube into the incubator with your inoculated tubes. It's a control. All right, we're going to make sure by doing that, you know, the next day that tube should still be red with no gas in it, because that it's not inoculated with anything. So it's a control, it's for comparison. Now take a look at the glucose tube. This looks just like the glucose tubes we saw with the other organisms. It's yellow, the media is yellow, and there's a little bubble of gas at the top of the Durham tube. So our Salmonella was able to ferment glucose and produce a gas. But if you look at the other two tubes, nothing happened there. We got growth in those tubes. We got growth, but there's no acid and there's no gas. So this particular Salmonella species is not able to ferment lactose, and it's not able to ferment sucrose. This is a very classic, typical result for Salmonella. Unlike E. coli, unlike Klebsiella, Salmonella is a little bit fastidious. Salmonella is a little picky. It's limited in what it can ferment. And it is a glucose fermenter, pretty much. It just can't make use of these other sugars. So if you're trying to grow Salmonella in the laboratory, it's just a little bit harder than growing E. coli, because Salmonella is a little pickier. It's a little harder to grow. So again, your third organism in your results table should be Salmonella. You can cross out where it says alkaligenes and write in Salmonella and record these results. And again, these slides, these results are going to be on Canvas. So if you can't record it right now in this instant, don't worry. You can do it at your leisure. All right, now let's take a look at our fourth organism, which is Pseudomonas. Remember, Pseudomonas was the one that we did in our disinfectant lab. We grew some Pseudomonas. We've also, we talked about how Pseudomonas is a common environmental contaminant. We talked about how Pseudomonas can be a problem in immune compromised people, and people like burn patients sometimes will get Pseudomonas infections. This is a nasty organism. Our Pseudomonas results look pretty similar to Salmonella, but there's one glaring difference. Yes, Pseudomonas is capable of fermenting glucose. It has turned the glucose tube yellow, but notice that there's no gas in the Durham tube. Pseudomonas ferments glucose using a different pathway than the Salmonella does. So there's no gas produced. That's important because it helps us differentiate between those two bad organisms. Salmonella and Pseudomonas are both pretty bad pathogens. And this is a nice way to distinguish between them based on whether or not they produce gas during glucose fermentation. The Pseudomonas cannot ferment lactose, and Pseudomonas cannot ferment sucrose. So we have an interesting set of results. Hold on, I'm just going back here. If I can figure this out. We have an interesting set of results for our carbohydrate fermentation tubes. So go in your lab manual onto the next page. If you have it, page 122. And this is where we'll record some data for our other two tests. We'll record data for this MRVP test and for our citrate test. Then we're going to look at two different organisms for both of these tests. We're going to look at E. coli again. And we're going to look at an organism called Enterobacter aerogenes. We've used Enterobacter before, so this is one that you've at least seen before. Hold on just a minute, and I'll see if I can get myself organized here.
Can you tell me what page that was again?
Yes, ma'am. We're on page 122. Sorry, I lost my chat box for a minute, but I've got it back. All right. So let's talk about this MRVP test. This is actually two tests that are run together. They're run as a pair. So remember, the MR stands for methyl red, and the VP stands for Voges Proskauer. Don't worry about that name. You can find it in your manual. So this is a test that is also about carbohydrate fermentation. It's just that we're looking at something a little bit different here. Remember, this is two tests in one, essentially. All right, let me make sure you're on. Yep, okay. All right, so the methyl red test has a different indicator in it than our carbohydrate fermentation test did. Remember, the carbohydrate fermentation test has an indicator dye called phenol red. It starts red and it turns yellow in the presence of acid. This test is exactly the opposite. Methyl red is a dye that starts yellow and will turn red in the presence of acid. Now, the reason they use methyl red for this test is because it has a little more of a range of color change. You can get anywhere from an orange color to a red color and have a positive methyl red result. Some organisms ferment and produce acid that is very acidic, and some organisms ferment and produce acid that is less acidic. Depending on how acidic it is, this product, you'll either get an orange color change or a red color change. And at the bottom of this slide in the notes section, I've got those details written down for you. So for now, understand that a yellow result is a negative result for methyl red. It means that the organism did not ferment and produce an acid. Oh, and I forgot to mention that the sugar in this test, this methyl red test, is glucose. So what this test is allowing us to do is not just say yes or no, it can ferment glucose, but what product is it making? Is it making one of the stronger acids, or is it making a weaker acid? So if you get an orange or a red with a methyl red test, it's positive. The orange means it's a slightly weaker acid. The red means it's a stronger acid. Now look at the Voges Proskauer test. That's the VP part of this test. Now this one is a little strange because in this test, what we're really testing for, let me get back there. What we're testing for is whether or not the fermentation product has a neutral pH. And this test is a little complicated because you have to inoculate the tube incubate it for 24 hours. And then when you take it out, you have to add a couple of reagents to it. And they talk about this in your lab manual. If you look on page, hold on just a sec. If you look on page 119 in your lab manual, they show you a chemical reaction. In fact, they show you a couple. They show you glucose at the top being fermented, again, first to pyruvate and then to either lactic acid or to this molecule called acetoin. There are different enzymes involved in making either lactic acid or in making acetoin. So again, we're looking at organisms that can ferment glucose here, but we're asking, what pathway are they using? Are they using the pathway that ferments glucose to lactic acid? Or are they using the pathway that ferments glucose to this neutral molecule called acetoin? Now, in figure B on page 119, they show us the two reagents that we have to add to the VP tube when it comes out of the incubator. We have to add a reagent called potassium hydroxide, that's the KOH, and we have to add a molecule called alpha naphthol. And what these reagents are going to do is push this reaction towards its conclusion. And the conclusion is this: if there's acetoin in the tube, the VP media is going to turn red, or I'm going to call it a brick red. It's not the red that you see in the MR tube, the methyl red tube. It's a dark red, almost like a blood red. And as you can see in the picture on the slide over on the right-hand side, it doesn't turn the whole tube red. You get a ring of red up at the top because you're dripping these reagents into that tube when you take it out of the incubator. So that's what we're looking for a positive VP test. We're looking for a red ring to form on the top of the tube when we take it out of the incubator and drip those two reagents in. So let's take a look at some results. The Methyl Red Voges Proskauer test is a classic test for differentiating between two coliform organisms, two organisms that grow in our intestine, E. coli and Enterobacter aerogenes. And these two organisms on this test have exactly opposite results. So let's take a look at the picture on the slide. You can see two methyl red results, that's the MR results, and you can see two Voges Proskauer results. Remember, the MRVP test is actually two tests. So look at tube A. The A tubes were inoculated with Enterobacter aerogenes. Tube A has a yellow color. Now, if this was a carbohydrate fermentation tube, we'd say, oh, that means fermentation happened and an acid was produced. But remember, this is a methyl red tube. Yellow means no fermentation here. Red means fermentation. So organism A, the Enterobacter aerogenes, had a negative result for its MR, its methyl red test. It does not ferment glucose. But look over, it does not ferment glucose to produce an acid product. Sorry, I should have said that better. Now look at the A tube over on the right for the VP test. There's our red ring at the top. Now this ring is a brighter red than the one I showed you last time. Again, most of the time when you run a VP test, this red ring is going to be really dark. But this picture happens to show a nice bright red ring. So this test is positive. This tells us that Enterobacter aerogenes, again, our first organism, when it ferments glucose, it makes a neutral product. It makes acetoin. So the MR told us that when it ferments glucose, it does not produce an acid. The VP test told us instead it produces a neutral product. Now look at E. coli. E. coli has exactly the opposite result. Over on the left-hand side, E. coli gives us a red MR result. E. coli ferments glucose and produces a strong acid as a result. And by strong, again, this information is in the slide set, but it's a pH of less than 4. Now go over to the VP test result. No red ring here. That means that there is no acetoin in that tube. E. coli ferments glucose and produces an acid. It does not produce a neutral product. So in your data table, at the top of page 122, you can now fill this out. Again, using these results. You can record that E. coli has a positive MR and a negative VP. And Enterobacter aerogenes has a negative MR and a positive VP, exactly the opposite. So we use the MRVP to help us differentiate those two organisms. But there's one more test we can use to differentiate those two organisms, and that's the citrate test. Now, the citrate test, I'm not going to get into the details of the chemistry right now. It's in these slides for you. When you go onto Canvas and you open this slide set, when you have a moment, under each slide there are notes, and I've got the details of the fermentation pathway of citrate in the notes for you. But what we need to be able to do is understand how to interpret this test, and that's what this slide is about. This agar, this citrate agar, comes in the form of either a slant tube, or you can also put it into a plain old agar plate. I see it more often in the slant tube, though. You don't need to grow a whole lot to get a result, so most people just streak a slant tube and then read it. And what we're looking for is, again, a color change. And the color change we're going to get in a positive reaction is blue. So I think the citrate test is one of the prettiest metabolic tests that we run, because we get a really pretty color change here. So on the left-hand side, this is how we would see a negative result. The agar stayed green. The slant tube stayed green. And on the right-hand side, we've got a positive result. Right on the slanted part of the agar where the cells would be growing, it has turned blue. All right, let's look at our results. Again, on this plate, we grew E. coli on one side, and we grew Enterobacter aerogenes on the other side. And these are our results. And it really does look like this, you guys. You really do get these amazing colors on this test. As you can see over here where it's green, this is the E. coli side. So it's typical for E. coli to not ferment citrate. It just can't do it. But on the right, here's our Enterobacter. Enterobacter aerogenes is quite capable of fermenting citrate. And when it does, the indicator dye turns blue. All right. So on the chat board, tell me if anybody has any questions about those test results, about the carbohydrate fermentation tubes, about the MRVP tubes, about the citrate test. If you have any questions, go ahead and type them in. Otherwise, I want you to turn to page 123. Page 123 while I answer some questions. So Kirsten is asking, how do you know if there's growth? In the pictures that I put for you in this slide set, you don't know. But I'm telling you that for all of our results today, you can record growth as occurring. As you can imagine, it's really important when you interpret metabolic tests that you first check for growth. If you pull a tube out of the incubator or a plate out of the incubator, that's a metabolic test, and you've got a color change and you don't see any cells growing on that plate, you've probably got a problem with the test. We always check for growth, okay? Because the only way that metabolism can be going on is if there are cells growing. So in order to trust our test results, we always check for growth first. Then we interpret the test, okay? So in your data tables, in your manual, you can check off growth for all of them. Now, at the top of page 123, oh, sorry, I have another question from Hannah, who says, what do we put for our control boxes? Okay, at the top of page 122, in the data tables for MRVP and for citrate, it asks you, it lists a control, and it asks for color. What they're asking for here is, what does a negative result look like? So remember, in the MR test, because methyl red is our indicator, a yellow color is negative. So our control is going to stay yellow. For the VP test, a negative is no red ring. No red ring. So our control tube should show no red ring at the top. Okay, good question. And down at the citrate test table, it also asks about the control. What color should our control tube be when we pull it out of the incubator? And the answer is, it should still be green. We should still see a green color in that tube. For citrate, if we get a positive result, that auger or that slant tube is going to turn blue. All right, now take a look at 123, page 123. You've got some questions to answer here. What I want you to do, though, at the bottom of the page is to cross out the critical thinking section. All right, I don't want you spending time on this critical thinking section because it's just not that applicable to these tests and why we do these tests. But I do want you to do the clinical application for this lab. Turn to the top of page 124, where it says clinical application. It says, you've put an opened bottle of orange drink into the refrigerator last week. Now the drink has bubbles in it and an off taste. Explain. So I want you to imagine that this is a bottle of some fake orange drink. So this is not orange juice. This is one of those, type of orange drinks that you buy that, literally is labeled as an orange drink, not orange juice, because there's no juice in it. Okay. So this is a sugary sort of fake orange drink that comes in a bottle. You have had a sip of it. You have had a sip of it, and then you put it into the fridge uncovered and saved it for later. And a week later, you came back, and not only is it suddenly carbonated, but it also tastes bad. And the question is, what has happened? All right. I want you to think about fermentation. Well, I want you to think about a couple things. First of all, if there are microbes in there, how did they get in there? What are the possible sources? And then how could a microbe have produced bubbles in there? What do you know about fermentation and gas? And why does it taste bad, do you think? Why might that taste bad? Okay. I want to give you a hint, something to look up. I want you to